Okay, well, hello everyone, and welcome to this research roundup session. In this session, we're going to be highlighting 3 research projects. We've got Dr. Tayal Paz, who's going to be talking about her project looking at DCM and the difference between men and women's hearts. We've also got Dr. Roberts joining us to give us an update on the brilliant HeartHive project. We also have a presentation, a pre-recorded presentation from Professor Elliott, looking at the work that he's doing with ARVC or ACM. We're also going to have some time to take your questions, and then I'm going to talk a little bit about some of the work that the charity has done and our own research plans. Okay, we're going to start off with a presentation from Perry. Like I said, Perry has pre-recorded this for us, but if you've got any questions about this project, then just post them on the Q&A and I can put them to him at the clinical Q&A later today. Brilliant. Please, we have Perry's presentation. Thank you.
It's now a great pleasure to be able to give you a brief update on research in the field of arrhythmogenic cardiomyopathy. The work I'm going to be showing you is funded by from a number of different sources, but predominantly from recent investments by the Arrhythmogenic Cardiomyopathy Trust. So to begin perhaps by just describing what the current state of the art is for arrhythmogenic cardiomyopathy. Now, this term has been applied to a fairly discrete entity described in the 1980s, which is a disease defined by structural abnormalities of the right ventricle, where we see enlargement of the right ventricle or local, what we call aneurysms within the wall of the right ventricle. And under the microscope, what we see is replacement of heart muscle cells by fat, shown in white, and fibrosis or scar tissue, shown here in blue. Now, this pathological substrate is matched by what we call an electrical substrate or abnormalities on an electrocardiogram, which are highly characteristic, and of course, more importantly, the presence of arrhythmia, what we call ventricular tachycardia, a rapid and potentially fatal arrhythmia arising from this abnormal right ventricle. Now, it's been known for many, many years that this entity, arrhythmogenic cardiomyopathy or arrhythmogenic right ventricular cardiomyopathy, often runs in families. And the first gene for this condition was identified on the island of Naxos in Greece. And here on the island, there are a small number of individuals who have severe ARVC associated with characteristic skin and hair abnormalities. And on the island, this is caused by a mutation in a gene called plakoglobin. Now, this is quite rare outside the island of Naxos, but what we now realize is that many people, if not the majority of people, with arrhythmogenic right ventricular cardiomyopathy have mutations in other genes that affect the same structure, which is what we call the intercalated disc that sticks adjacent heart muscle cells together. And indeed, actually, as time has gone on, we've identified quite a large number of genes that affect the structure, but also other genes which affect the heart muscle and cause what we call a phenotype, an appearance within the heart, very similar to that seen in classical ARVC. Now, the main problem that is associated with this condition is that of sudden cardiac death, often in young people. The prevalence of this in registries of deaths amongst young people or athletes report ARVC various percentages, from 4% in the United States up to a quarter of all deaths in Italy. If we look at data from the United Kingdom, then best guess is maybe about 1 in 10 deaths in young people caused by ARVC. But I think sometimes there's a sense that, well, that's okay because we have really good therapy for this, the defibrillator, which we know can be life-saving. As you can see here in this Italian study, patients who received an ICD, virtually no mortality compared to those who continue to have fatal ventricular arrhythmias without a defibrillator. But of course, the problem is that many people are not identified with the condition. And that may be because actually when we do an ECG or take a picture of the heart, they don't have the classical features of ARVC. In some patients, the diagnosis is overlooked because the patient may have not the classical features, but perhaps just scarring in the left ventricle, which is otherwise functioning normally, Or at the other end of the spectrum, it may be people presenting with severe heart failure, where the diagnosis of arrhythmogenic cardiomyopathy is now overlooked because of the priority of managing the heart failure problem. So I think if you look at the key questions that still need to be answered in arrhythmogenic cardiomyopathy, it's how do we improve early diagnosis? How do we better stratify people to identify those individuals who are most at risk from both sudden death and heart failure? And how do we prevent the disease from progressing? So the project in which ACT is invested is built around the whole concept of personalizing the approach to therapy. This is a concept which is very prevalent in medicine now, particularly in cancer, where we try to better understand the underlying cause of disease and then develop new therapies targeted to those causes. But as in this editorial in Nature from a few years ago noted, personalized medicine in cardiovascular medicine is still a little bit behind the ball. And so our aim in this context of arrhythmogenic cardiomyopathy is to develop a research platform that allows us to better understand the mechanisms of disease and ultimately to develop new therapies which target those mechanisms. So the strategy that we're employing is to use the large patient cohorts that we see at our own institution, but also other institutions in the UK and around the world, and to take from those individuals what we call biosamples, blood, urine, and maybe other tissues, and then to take those to the laboratory where we can extract from them DNA or RNA to study the underlying genetic code and its abnormalities, but also how those abnormalities in the genetic code result in abnormal proteins and in the activation of other abnormalities in the body, for example, inflammation. Fundamental to this exercise is the development of a robust database which allows us to collect and share information between investigators. And this has now been made possible by investment from ACT and other charities. And we're using what's called a REDCap database, which allows us to collect in great detail the characteristics of patients, their presentation, and their natural history. We're being able now to develop pathways where we can see people in the clinic, inform them about the research projects that are available, and with their consent to take their data and then to anonymize those data and then use it for these various different research pipelines that I've described. The value of doing this, of course, is that it allows us to do this at scale, but also in line with current guidelines and recommendations for the preservation of anonymity of data. The overall schema for the project over the next few years is to improve our characterization of patients, to look at genetics, protein expression, and also to develop models of the disease using cells and sparingly animal models, and to use this information to develop new risk models for the disease, to discover and validate markers which tell us about the diagnosis, but also likely prognosis of individuals, and of course, ultimately to develop platforms for development of new drugs. And the net result of this is we want to increase awareness and earlier diagnosis of arrhythmogenic cardiomyopathy, to reduce sudden deaths, but also to prevent heart failure. And of course, the ultimate goal is to cure the disease. Work in progress. We've been working with colleagues at St George's Hospital to develop cell models, which allow us to insert genetic abnormalities that we see in humans, and to then understand how that affects the cells in the heart, and to use that knowledge to develop therapies which can prevent the development of disease. Sometimes we have to use animal models for this purpose, and we're also working with a number of different colleagues using mouse models of the disease, and again using this as a platform for exploring novel therapies to prevent disease development. One of the key areas that we're exploring is the role of inflammation in arrhythmogenic cardiomyopathy, and this is something which has been noted for many years. If you look at the tissue taken from hearts of people with arrhythmogenic cardiomyopathy, we often see little areas where there are inflammatory cells. And indeed, some people, particularly in the early stages of the condition, may present with clinical features of inflammation in the form of chest pain, arrhythmia, and markers in the blood which suggest that they have inflammation in their heart. And these episodes, what sometimes are referred to as hot phases of the disease, are often the moment when the disease becomes manifest and are also associated with disease progression. What we've been able to do is to try to study inflammation non-invasively using different imaging techniques, and in particular one called PET scanning, which allows us to identify areas of inflammation in the heart of people presenting in this hot phase. The other work that we're doing with colleagues at Queen Mary University in London is to try to identify in the blood abnormal white cells that may be responsible for initiating and maintaining inflammation in the heart. And we can do this by extracting white cells from peripheral blood and to look for markers on those cells which tell us whether the cells are active and whether they're directed against heart tissue. A piece of work that illustrates the value of the databasing and also of international collaboration is this recent work where we've tried to evaluate a proposed model for estimating risk in ARVC. And what we've been able to do in this model is to confirm that the model does indeed predict risk, but that it's critically dependent upon the underlying genetic abnormality. And also some features of the disease which have been proposed as markers of risk, we're also showing that these are also dependent on the underlying genotype, pointing the way to actually risk models built around genetic diagnoses rather than just simply the clinical diagnosis of arrhythmogenic cardiomyopathy. So I hope that gives you a flavor for what's happening in this field. I think it's really exciting. I think we're already seeing progress in our ability to characterize the disease, to predict its outcome. But I think in the next few years, with the help of charities such as ACT and Cardiomyopathy UK, I think we're going to see the development of new therapeutic options. Thank you very much for your attention.
Okay, well, thank you. Like I said, that was a pre-recorded presentation from Perry. Perry is going to be joining us later today for the Q&A session. So if you do have any questions about that presentation, just put them in the Q&A and then we'll pass them over. We're now going to move over to Ang Roberts, we're going to start off with a presentation about the HeartHive, and then Anne's going to be answering any questions that come out of that. Brilliant. Okay, can we share that presentation, please?
Hi, I'm Ang Roberts, and thank you for the opportunity to talk to you today about the HeartHive. The HeartHive is a web portal to connect research-willing participants with research opportunities, developed in collaboration between Imperial College London and Cardiomyopathy UK. And I'll introduce the Imperial team to you. There's myself, Ang Roberts. I'm a consultant in clinical genetics at Great Ormond Street Hospital. Dr. James Ware, a consultant in cardiology at the Royal Brompton Hospital, and Rachel Buchan, our project manager and genetic sequencing guru. Now, the HeartHive is open to anyone with cardiomyopathy or myocarditis, or with a family history of these conditions, or anyone found to be carrying a cardiomyopathy causing gene variant. We have over 500 registered participants so far, and you can join us by heading to the hearthive.org. Here's a screen grab from our website where you can see where to register or watch a video with more information. If you click on our FAQ tab at the top of the page, you'll reach a frequently asked questions section with more information on how to register, what happens next, and what research is available. Now, the HeartHive all came about following a patient engagement event where we heard you telling us that you really want to get involved with research, but it can be difficult to access research opportunities and to get involved. We know that participating in research has benefits for patients, either through participating in clinical trials or through being proactive in tackling your condition. We also know, and we hear you telling us, that it's important to allow patients to input into the research agenda to ensure that we're addressing questions that are important to patients. This chart shows the participants in the HeartHive to date, and you can see the pink bar at the bottom of the chart shows that we have a large number of participants with dilated cardiomyopathy, and the orange bar represents participants with hypertrophic cardiomyopathy. The blue bar represents our cohort with no heart disease, who are acting as a control group for some of our studies. We also have a broad range of other cardiomyopathy diagnoses, some people with a family history of cardiomyopathy, and some with myocarditis. We're running a number of studies and surveys through the HeartHive with more to come, so we have something for everyone. Our first study is the HeartHive Cardiomyopathy study. This was a proof of principle study to demonstrate that this approach of doing research through our web portal and remotely is effective. It's open for anyone with hypertrophic cardiomyopathy or dilated cardiomyopathy. We ask you to sign up online through the web portal and to complete an online health questionnaire and a shorter questionnaire at the end of each year. Through the post, we'll send you a kit to collect a saliva sample for DNA and genetic studies, and we'll ask for access to your electronic health data through NHS Digital. We have over 200 participants in the study now, and more than 80% have completed their first yearly follow-up survey online, which is an incredible level of engagement. The chart here is showing that we have a lot of participants from both dilated cardiomyopathy groups and hypertrophic cardiomyopathy, and we have a good range of both men and women. The pilot phase of the cardiomyopathy study is wrapping up now, and genetic results letters are being reviewed for those who've opted in to receive them. We found that the results of our research genetic analysis closely match with what our participants are reporting from their genetic testing within the health service. The chart here is showing our results for participants with dilated cardiomyopathy in pink on the left and hypertrophic cardiomyopathy in orange on the right. You can see that for both groups, most participants did not have a significant genetic cause for their cardiomyopathy identified. On the right, that you can see that for our hypertrophic cardiomyopathy group, we identified variants in 3 different genes as being significant. On the left, in pink, for our dilated cardiomyopathy group, you can see that we identified variants in 4 different genes. Now, there may be some participants watching who have reported to us that they have a gene change in the FLNC gene, which is not shown here. That's because that gene is not included on the panel of genes that we're analysing during our pilot study, as it's a relatively new gene. As we wrap up our pilot study and look at the results, they're showing us that this form of research is effective. And so further funding has been secured to extend this study past the pilot period. So we'll be continuing to recruit participants with dilated or hypertrophic cardiomyopathy, and we'll also be extending the genes that we're studying, looking at all genes going forward. Our second study is the HeartHive COVID-19 study. This was driven by the HeartHive team together with Dr. Daniel Hammersley, a PhD student at Imperial and the Royal Brompton Hospital. Using the HeartHive, we were able to respond quickly to the pandemic and to safely gather information from people with cardiomyopathy. We asked about your health, your psychological well-being, advice you received, and interruptions to normal cardiac care. Participants have been completing surveys every 3 months since mid-2020. And from our study, I'm showing you some results here for the cardiomyopathy group in orange and the control group with no heart disease in blue. And you can see that participants with cardiomyopathy were far more likely to report a deterioration in their physical health during the pandemic. Interestingly, we found that participants with cardiomyopathy were slightly less likely to report COVID-19 symptoms. We also asked our participants with cardiomyopathy on how the pandemic was impacting their clinical care. We found that about 1/3 of you reported a missed clinical investigation or a rescheduled outpatient appointment. About 1/6 of you reported a cancelled outpatient appointment. Smaller numbers reported postponed or cancelled clinical procedures and missed medication doses due to the pandemic. Importantly, 40%, so nearly half of you, reported that your needs are not being met by telemedicine. We also asked both our cardiomyopathy group and our control group without heart disease how the coronavirus makes you feel. And both groups reported some feelings of stress, depression, fear, or helplessness. Our participants with cardiomyopathy were slightly more likely to feel stressed or fearful, but interestingly, less likely to feel helpless. The results of the HeartHive COVID-19 study have been combined with data from the Royal Brompton Hospital and NHS Digital and written up for publication. They're currently in review with a medical journal and will be published on our website as well as soon as we get the go-ahead. We hope that insight provided by this study could help to prioritise the clinical needs of patients with cardiomyopathy and in turn guide adaptations to clinical services in order to meet these needs. Another important aspect of the HeartHive is patient engagement, doing research in partnership with you. The HeartHive provides us a platform to directly ask our participants for input and opinions on research proposals, helping us to take into account patient concerns before we design a study protocol. Dr. Brian Halliday, a newly appointed consultant in cardiology at the Royal Brompton Hospital, has been asking participants with dilated cardiomyopathy for input into his latest research, with a short survey about medication. He's had a fantastic response rate compared to other methods used in the past to gather patient opinions. He can now address more impactful research questions thanks to your responses. So what does the future hold for the HeartHive? We're working hard to bring our participants the opportunity to register their interest in clinical trials and to undertake a short survey to check their eligibility. We're in the final stages of negotiation with a clinical trial currently recruiting in the UK, so watch this space. We're extending the cardiomyopathy study. We're continuing to recruit participants with dilated or hypertrophic cardiomyopathy, and we're collecting samples by post. We'll be looking at all genes going forwards. We're continuing the COVID-19 study, sending out surveys every 3 months. This study is open to anyone with any form of cardiomyopathy or to a control group with no heart disease. You can participate in the control group even if you have diabetes or high blood pressure. So, if you have friends or family who are unaffected but want to contribute to cardiomyopathy research, this is a study for them. We'll be adding more new studies soon, so keep checking back. So it just remains to say a huge thank you to everyone who's participated so far in the HeartHive. If you haven't, please head over to our website, thehearthive.org, to find out more and to register. Please spread the word on Facebook, Twitter, and Instagram, and our handles are shown here on the right of the page. And we're also available on email, so you can reach out and let us know if you have any suggestions or questions. Contact info@thehearthive.org. Thank you.
Okay, great. Well, Thank you, everyone. We've actually got Ang here there, so there's a couple of questions that have popped up that we can address now, and some that were sent in previously, Ang, if you're happy to cover them off. There's one question that we're wondering about. Obviously, we talked about how COVID has affected people with cardiomyopathy, but how has COVID actually impacted on research and research efforts?
Well, in terms of the HeartHive, it hasn't impacted too much. We did have to pause saliva sample collection during the peak of the first wave until we could be sure that it was safe for our participants to be heading out to the post box to send them back to us, and also to confirm that it was safe for us to receive them, and that the liquid that's in the tube will inactivate an RNA virus like the coronavirus. So really, ever since then, we've been continuously doing our research and collecting the data.
Okay, brilliant. Thank you for that. And there was another question I thought we can address now, for, I suppose, both for you and for Paz, which is, what do you think is the biggest change you've seen in cardiomyopathy treatment, and what do you think the biggest hope is for the future? Paz, do you want to start off with that one?
Yeah, I think it's a really, really exciting time to be treating patients with cardiomyopathy, and I think the next 5-10 years are going to be, we're going to see huge changes. I think things that are really exciting are the advent of gene therapy. So if you have a genetic form of certain conditions, I think there's real hope, and there's really active progress now that we may be able to correct and even cure some of these conditions in the future. It's been demonstrated as a proof of principle with a form of amyloid, which is slightly more straightforward to get to. But, we've all seen, all of us have seen in the last couple of years how much science can rapidly progress. So personally, that's what I'm really excited about. And secondly, also for patients with heart failure, so dilated cardiomyopathy or hypertrophic cardiomyopathy that then leads to sort of a heart failure type picture. There are so many more drugs with really with benefits both life-saving and also to improve morbidity, so quality of life and how patients are doing, which again, 5-10 years ago, we had just a tiny proportion of tools up our sleeve. So it's a really exciting time.
Brilliant. Any thoughts?
I would echo what Paz has said. It's a really exciting time, and I think things like gene therapies are really exciting to see coming through. I think similar to that are therapies that we can identify and target according to the underlying genetic diagnosis that aren't necessarily a gene therapy, but address the underlying biology that's causing the heart disease, rather than just treating the symptoms.
Okay, brilliant. All right, we'll try and address some more questions after the next presentation, but we're going to move now on to your presentation, Paz, if that's all right.
Super.
I'm going to share my screen.
Thanks so much, Joel, and everybody for inviting me to be here. So my name is Paz Tayal. I'm a cardiologist and an academic at Imperial College and the Royal Brompton Hospital, London. So next slide. So I know dilated cardiomyopathy probably needs very little introduction to this audience, but in case some of you are new to our community, dilated cardiomyopathy is one of the leading causes of heart failure. These are cardiac MRI pictures that you can see on your screen. The normal heart is shown in the small box, and a dilated cardiomyopathy patient is shown in the large box. And as you can see with dilated cardiomyopathy, the heart gets bigger, baggier, and just can't pump blood as effectively as a normal heart can. So this means that blood backs up and fluid builds up in the lungs and the legs, and it leads to symptoms of breathlessness, swollen ankles, fatigue. It affects up to 1 in 250 people. Next slide. And unfortunately, despite treatment, outcomes really vary. Some patients do really, really well and completely recover, have no symptoms and live a completely normal life, yet other patients go on to need heart transplantations. So it's clear that our approach to treatment, this sort of one size fits all, treat everybody the same, isn't really working. We really need to improve our understanding of the condition to make outcomes better. Next slide. And there's work that I've done and some pilot data and observations that I would like to share with you today, which is why I think that studying sex differences, so the differences between males and females with dilated cardiomyopathy, really offers a scope to improve outcomes for all patients with the condition. Next slide. So number one is that dilated cardiomyopathy affects twice as many men compared to women. Now, by itself, that's not necessarily that interesting. We see this quite commonly in lots of other conditions, and we also see this with certain forms of heart disease, such as coronary artery disease. But what is more interesting and noteworthy is that if you look at certain genetic forms of the condition, where for reasons of inheritance, you would expect 50% of patients to be male and 50% to be women, you don't see that. You still see the condition affecting more men than women. So that tells you one of two things. Either men are more vulnerable in some way to the condition, or women are protected in some way. Next slide. The second is that despite appearances, men's and women's hearts are very different. So my work and other data show that men's hearts are bigger, have more scarring, and don't pump as well compared to women's hearts. And that's in dilated cardiomyopathy. But even if you look at completely healthy people, even down to prepubescent teenagers, you still see clear differences between male and female hearts. Next slide. But despite this, despite the fact that women with dilated cardiomyopathy seem to have a milder heart condition, actually, and this is my data here, women with the condition actually have a worse outcome. So this is something called a survival curve. And you can see women are represented in red and men in blue. And the higher up the chart you go, the more adverse events, so complications of your condition that you have. And you can see that whilst men and women are the same at the end of the study period, so 5 years, certainly women have more complications of their condition early in the disease. Next slide. So despite all of these observations, the role of biological sex, so that is being male or female, is very poorly understood in dilated cardiomyopathy. Women undergo or experience a number of very unique life events, pregnancy, menopause, that men just don't go through. And we don't know what the contribution of those conditions to developing dilated cardiomyopathy and also its natural history are. And despite all these differences between men and women's hearts, there are no sex-specific either diagnostic thresholds or treatment thresholds. Right now, we treat men and women exactly the same. Next slide. So these, I think, really highlight the need for sex-specific research, which is what I would like to do. Next slide. And so my research aim is to evaluate sex-specific differences in dilated cardiomyopathy. This is a project that I have sought, I'm really grateful for input from Cardiomyopathy UK and patients from Cardiomyopathy UK into planning this research project. And there are 3 main studies that I would like to do. Next slide. First is epidemiology. So really looking at what extent sex-specific variables determine how dilated cardiomyopathy manifests. And this really means looking at dilated cardiomyopathy in the context of unique female-specific factors, and that includes menarche, menopause, pregnancy complications, pregnancy losses. And some early data that I have suggests that menopause may be an as yet unrecognized cardiomyopathy risk factor. So one potential impact of this piece of work would be early detection of at-risk populations. So perhaps a woman who is perimenopausal or going through the menopause with dilated cardiomyopathy is at increased risk of complications and needs closer surveillance. And that's something that I'd like to explore further. Next slide. The second piece of work I'd like to do as part of this project is to look at risk stratification for sudden death in dilated cardiomyopathy. And here I've built a collaborative consortium across Europe, North America, Australia of 9 centres with 6,500 patients with dilated cardiomyopathy. And I'd like to build something called a risk prediction model. So that will a series of algorithms that try and tell us who will do well and who will not do so well, but one that works really well in women. Currently, the life-saving cardiac defibrillators are recommended in patients, men and women, once you get to a certain threshold of heart function. But again, some early data I have suggests that we actually should be using different thresholds in men and women because their hearts are different. And so one potential impact of this work is that more women may get appropriate treatment with life-saving defibrillators. Next slide. And the final bit, remember how I said that the structure of men and women's hearts are very different. So I'd like to look at the molecular basis of why this might be and look to see whether there are any differences in the genes that are differently switched on in male and female hearts, both in health and also disease. And again, some of the early pilot data I've got suggests that there are differences, particularly in cardiomyopathy genes. And so hopefully we can use this data to try and work out why there are those differences in heart structure that may then help us work out novel treatments in the future. Next slide. So I think who benefits from this research? Well, I think clearly patients, clinicians and researchers studying dilated cardiomyopathy and heart failure, both in the UK and globally. But I think it's also relevant to academics and clinicians in related fields. So anybody with an interest in women's health, women's health extends well beyond the bikini line, and it's all the organs in lots of different ways. Clinical trialists, we know that women are currently very underrepresented, particularly in cardiology trials. So treatments are tested in men and then applied in women. I think this work will be of interest to policymakers, particularly the work around defibrillator treatments and also industry, because we have to be able to target the right treatment to the right patient at the right time. Next slide. So I really just want to thank you for the opportunity to share, some of my early data with you, my science, what I'd like to do. A huge thanks to the patients who've supported this research so far at Cardiomyopathy UK, and I'd be happy to take your questions either now or come and find me on Twitter. Thanks.
Brilliant. Okay, thank you for that, Paz. That's really appreciated it. We've got a few questions that have come up already, if you're happy to answer them. I think the first question is really just if you can talk a little bit more about how the sort of input that the sort of focus groups that we put together and the sort of patient input has actually shaped your thinking with this project.
Hugely, really informative. So one thing that you have to do when planning research studies, you come up with a hypothesis and you work out how to test it. And so you have a question that's framed in a certain way. And one thing that really strongly came out of the focus groups was that the way that I was looking at the issues, it felt like I was just interested in women and women with dilated cardiomyopathy, and the men didn't matter. But actually, this work stands to improve outcomes for all patients with the condition. And so really making sure that that comes across both in the study design and in the way that I talk about the work was really important. And the second thing, people were very generous in sharing their experiences. And one of the themes that kept coming out of those focus groups is that women of a certain age, so those who were peri and postmenopausal, had symptoms for a number of years, and they certainly weren't taken as seriously as the symptoms that the men in the groups had. And it took them a lot more years to get their diagnoses, suggesting that we really potentially need to do a bit more work looking at how women get symptoms when they have this condition, and if that differs to how men get symptoms.
Okay, that's interesting. And another question, if you don't, if you can both address this one, if that's right. Paz, you touched a little bit about what the implications of the work, particularly around ICDs. And can you just talk a little bit about what you hope to be the outcome of the work and how you see that as directly improving things for people with cardiomyopathy?
Which piece of work, John?
So the HeartHive project as a whole.
So I think one of the main things is to make research more accessible and make it equitable, so that wherever you live in the country, whichever hospital you attend, that you have an equal opportunity to take part in research, that it doesn't require you to be at a big university centre, it doesn't require you to travel really long distances for a lot of study appointments. You can do it from home, get online, we'll send you all the stuff from the post. You can take part in as many surveys as you want. So I think that's going to be a really big outcome from the HeartHive. And second, I think patient engagement, that we have the opportunity of, as we've demonstrated and as Dr. Brian Halliday has taken advantage of, to ask our participants for their opinions directly through the platform, and to have that inform research and find out what's important to participants and how it's affecting them. So something like the COVID-19 study that we put on, we're able to launch it really quickly and get really direct data about how the pandemic was impacting patients with cardiomyopathy, and particularly impacting their access to their cardiac care and how that was impacting them in general.
I think, if I can say, I mean, from the charity's point of view, so it's a project the charity has always been really keen to support, because I think we see it as a way of removing barriers between people who want to be involved in research and researchers. So it's easier for people to engage with research, and it's quicker for researchers to get the information and the data that they needed. And our view was that, by investing in this sort of infrastructure for research, we'll in time be able to encourage a lot more people to come into the field of cardiomyopathy and be able to access the data and the people with cardiomyopathy that they need for their work. Paz, one of the things you mentioned in your presentation was about ICDs and risk. Can you just talk a little bit more about how you think your work might affect that?
Yeah, so it's quite a controversial area now in dilated cardiomyopathy. So the guidelines still state that patients with an ejection fraction, so a measure of the pumping function of the heart below a certain limit, should be considered for an implantable defibrillator. The idea being that that's life-saving therapy should they have a very dangerous heart rhythm, so risk of sudden death. Big trials have come out though, showing that actually the benefits, now that we've got so many heart failure treatments and we're treating patients with more drugs, better drugs, and putting in special types of pacemakers in patients, are actually reducing the amount of benefit we see from the defibrillators. Nevertheless, there are still lots of unanswered questions, and clinical practice is still in patients who we think are appropriate to put in defibrillators. But the paradox here is that there are patients who get defibrillators put in as like a life insurance policy almost, and then they never go off, right? And so, for the patient, that is in some level good, but also equally they've then gone through a procedure that they may not have needed. And equally, there are patients who currently are not eligible for a defibrillator, who then unfortunately experience severe complications of their disease, have a cardiac arrest, and would have benefited from a life-saving defibrillator, who at the moment are not eligible. So what I want to look at, just as a small piece of that puzzle, to see actually, are men and women different? And those structures, those different structures of the heart that we see in the way that heart function is very different in men and women. Do we need to rethink the way we think about defibrillator therapy in women? Are we missing a whole proportion of women who would be potentially eligible for this treatment? And the answer may be not, but I think it's a question that we really do need to look at.
Brilliant, thank you. There was a question about HRT, but I think what I'll do is I'll pass that over to the general clinical, because I know we're about to run out of time. So with the time left, I do just want to quickly talk about our involvement in research. And I suppose that there's two sorts of research activity that we undertake as a charity. Firstly, similar to how we work with PAS, but providing insight to clinical researchers. And this is where we work with researchers to develop their own projects, and often means bringing together focus groups or conducting surveys. And we've really seen how that insight shapes the work of these projects. I think a big example of this is that over the last year, we've been working with this international team led by Oxford to bid for a £30 million grant from the British Heart Foundation. This is called the Cure Heart Project, you may have heard about. We've still got our fingers crossed to see how successful that was been. But our involvement in that really did help shape the work, make the work more broad in its scope, and made it easier for individuals to access. I think the other thing that we do is that it's important to remember that not all research is clinical, it's not all test tubes. There's quite a bit of work that the charity does itself, which we would suppose call social research, by surveying our own community. And again, this is something that we use to shape NHS services and help get a better deal for people with cardiomyopathy. Recently, for example, we ran a large survey regarding the difference between face-to-face appointments and remote appointments, so Zoom calls and phone calls. And it's been really useful to get the insights from people with cardiomyopathy and to be able to feed those back to the clinicians who are struggling to work out how to manage their backlogs, and be able to tell them some insightful information about which sort of individuals may be happier to take remote calls rather than face-to-face. So there is a lot of research work going on at the moment, and it's something that we really do hope to increase over the next year. And I suppose a plea from me is that, do look out on our website for opportunities to engage with research. Do sign up. I think from next year we'll be looking for people to volunteer to be joining a rapid response panel. So we do encourage people to sign up for that. And we are running out of time now, so that's it. I just want to have a big thank you to Paz and Ang, and of course to Professor Elliott. And if you have any questions about our research, either put them in the Q&A or do get in touch with the charity afterwards. Thank you.
Thank you.